Thank you very much for sharing your morning with me here today. I am super excited to share not just what I do at Mote, but what about 30 different staff members and 5 different PhDs really focus on within Mote's infrastructure, and that's Coral Reef Research and Restoration. And I'm happy to be back here because being in Boca Grande, I think it was exactly Coffee with the Scientist 2 years ago was my last in-person presentation outside of Mote's building prior to COVID. So it's kind of come full circle for me, and I'm really excited to be able to be back here and give you all some updates, or for those of you that haven't seen this presentation before, share with you all this amazing research that we're doing here at Mote to help save our coral reefs. So as I mentioned, at Mote alone, we have about 30 different staff that focus on coral reef research and restoration. And we also have huge amount of infrastructure and finances that support all of the research and the restoration that goes on within this kind of initiative. And several of us are here located in Sarasota that really focus on studying some of the major research questions associated with our activities. But the bulk of where this magic happens is down in our Summerland Key facility, our Elizabeth Moore International Center for Coral Reef Research and Restoration. This is truly an international hub where scientists and restoration practitioners from around the world come to collaborate with Mote scientists, conduct their independent research, and learn how to do restoration, not just here in Florida, but literally around the world. This is a 19,000 square foot piece of infrastructure that contains classrooms, lodging, wet lab space, dry lab space, and that's pretty impressive in itself, but it also contains the world's largest land-based coral nursery. So although it's difficult to see in this image, to the left, under those black tarps and beyond, is actually our land-based coral nursery. We hold thousands of corals that we grow and propagate for restoration purposes. And then just offshore of this facility, about 7 miles underwater, we have our coral nurseries as well. And I'll show you what that looks like as if you were there today. So why do we put so much of our time, people power, and infrastructure for coral reefs, for research and restoration? And it's because this ecosystem is so incredibly important. It is one of the most biologically diverse systems in the world, but also the most biologically diverse ecosystem in the ocean. It's estimated that 25% of all marine life rely on coral reefs at one stage or another in their lifespan. And so it's a critical ecosystem for biological diversity. But this ecosystem also supports what we call ecosystem services to humans. So whether you realize it every day, whether you realize it or not, every single day, humans around the world are benefiting from coral reefs. That is because they are an incredible source of wave attenuation and absorb wave energy, protecting our shorelines. So like Florida's coral reef, which goes from Martin County all the way down to the dry Tortugas, if it wasn't there, we would have a huge amount of erosion of our infrastructure and our shorelines along the coast within that area. This helps to protect thousands and thousands of people's homes, our roads, our hospitals, our schools, because as storms roll through, that wave is getting broken up on the coral reef, and it helps prevent erosion and flooding from occurring in our nearshore environment. These ecosystems also provide a novel source of medicines that we use to fight human diseases. This can include antibiotic-resistant bacteria, cancer, and even Alzheimer's. So today, there are chemicals that only exist in our coral reefs that we harvest and utilize to fight some of the major concerning diseases that we're fighting today. And finally, it's incredibly important for food stability, income, and jobs for people globally. It's estimated that at least 1 billion people around the world rely directly on coral reefs for their income and stability and food source. And to bring it back home to Florida, Florida's coral reef are a huge economic engine and the foundation of our economy in Florida. That reef helps to attract 16 million visitors to the state, 6 million of which want to go explore directly the coral reefs within the area, helps to support over 70,000 local jobs, whether that's within the tourism industry or the fishing economy. And it's estimated that they're worth at least $8 billion to our state economy, providing over $2.5 billion in revenue every single year to our local businesses. Incredibly important for economic stability here. But unfortunately, we are losing coral reefs at an unprecedented rate because the critical organism that creates the reef and the habitat, the corals themselves, are dying. This is Carey's Fort Reef, which is a reef in the northern part of the Florida Keys just a few decades ago. And you can see it's teeming with these stony corals, the staghorn coral in the front and the elkhorn coral in the back. These two species are now listed under the Endangered Species Act because there are so few of them in existence today. And now if you go back to Carey's Fort Reef, you see the ghosts of these corals that have passed. The skeletons remain, are getting degraded, and we're seeing algae blooms, very little fish, and certainly no stony corals. So if you imagine that this room is Florida's coral reef, in the 1980s, half of it would have been covered by stony corals. Now only 2% of that reef is covered by stony corals, and they are not the ones that are providing the critical habitat and ecosystem services that are essential for this ecosystem to be functioning. However, there are organizations like Mote, and Mote has been leading the charge to bring back these coral reefs away from this slippery slope to slime to the way that things were decades ago, using innovative coral restoration science. And we started this over a decade ago with our in-water staghorn coral nurseries. So if you go to the Summerland Key campus and just drive straight offshore about 7 miles and go 20 feet underwater, this is what you will see. Acres of these underwater coral trees. They're made up of PVC pipes and floats, and instead of pine needles dripping off of them, like you would see in a coniferous tree, you have these branching corals that are dripping off of these infrastructure trees. And we have hundreds of them within 2 nurseries, one right off of Summerland and actually one off of Key West. And those nurseries hold at least 20,000 fragments of staghorn coral at any given point in time. They represent over 200 different genotypes. That's really important because we want genetic diversity in our restoration plan. All of us in this room, we're different genotypes because we all have different DNA. And our DNA can influence how we fight diseases, how we deal with nutrition, whether we can withstand stress. And so it's really important that genetics become a part of the conversation when you're thinking about conducting coral restoration. Now, how do we do this? We outplant about 20,000 of these staghorn corals every single year, and we outplant them in what we call monoclonal clusters. So we can take a diver, swim right up to one of these trees with some clippers, and clip off a branch tip. Now we're leaving the broodstock there so that it can grow back. So it's a sustainable cycle. So you can actually go get these branch tips off of each tree and then bring them out onto the reef in these underwater baskets. We take the branches and we outplant them in a cluster of 5 using a masonry nail that we just hammer into the reef structure and tie the corals onto the masonry nail with a biodegradable tie. The corals actually grow over the ties, over the nails, and attach to the reef within about a few weeks, maybe a month. Now that's really important because that increases their stability that they have onto the reef. It makes them a permanent reef structure. And then within just about 8 months for this species, you get this growth that you see on the right, where all of those 5 individual fragments are now blooming, they're touching each other. And because we've thoughtfully integrated the genetics, we know that those 5 that we put out all came from the same original colony. They have the same DNA. That means when they touch each other, they don't fight, they fuse. And so this picture on the right is a fused coral thicket of one genetic variety, and it is now of the size that's going to reproduce during the next spawning cycle. So now corals reproduce based on size, not age. So we have reached that reproductive size within about 8 months of this particular species. That means the next spawning cycle, they're starting to release eggs and sperm to the water column that can eventually settle and help us out in that restoration process by having self-sustaining reefs, by putting these corals out there that are going to be providing new babies for the next generation. And we get really good survival. So over the last 10 years, we've figured out how to grow these corals, how to propagate them, and how to outplant them onto our degraded reefs of Florida to get over 95% survival within that first year after outplanting. That means that they're not dying from the transportation stress, they're not dying from shock associated with getting out there. They are there and they are establishing themselves into the reef community. Now, we don't just focus on staghorn corals. That's where we started because those were the easiest to kind of grow and hack the biology from. But we want to do community ecosystem restoration. We need to tackle all the different corals that are out there, the boulder corals, the brain corals, and the branching corals. But the problem with some of these other corals, the boulders and the brains particularly, is that they grow so slowly. The staghorn coral grows about 10 centimeters a year in every direction. So you can get a lot of growth really quickly, but the boulder corals only grow a couple millimeters a year. And to reach a reproductively sized boulder coral, it would take decades to create through one larvae and just wait for it to grow. These corals were always termed the orphan corals of coral restoration because nobody wanted to deal with them because you could barely see any success within the life cycle of a grant or a graduate student. And so they were kind of always left to the side. But our scientists took the bear by the ears, bull by the horns, there we go, and said, okay, let's figure out how to hack this biology of these corals and get them to grow faster. And through some trial and error and some aha kind of accidental moments, we created this process called microfragmentation and fusion, or reskinning. Now, the process of microfragmentation is taking a larger piece of a coral. It can be, basketball size. It can be the size of a silver dollar, and cutting it up into tiny individual sized pieces that are just like 1 centimeter square. So if you can see on those discs right there, those are ceramic discs that the corals love to grow on. They basically mimic reef structure. And on each of those discs is a coral colony we've created through the process of microfragmentation. But these corals are actually really big. They're about ready to go outplanting. The process of microfragmentation can take one of those discs and cut the corals up into maybe a dozen smaller pieces, where each circle you see is an individual polyp of the coral. That's all that's needed to regrow a coral from a cutting, basically, to a new organism, is one polyp. So we take bandsaws and we have our staff. We have an army of interns that go through our broodstock and cut them up into microfragmentations, tiny little pieces that are then reattached to these ceramic discs and allowed to grow out in our land-based nursery on the Summerland Key campus. When the corals are the size of a disc, so about that $50 size, they're ready to go out onto the reef. And depending on the species, we can get those microfragments to grow to be outplantable size anywhere between 4 months and a year, depending on the species. But that process of microfragmentation makes those corals grow 50 times faster. So we're not having to kind of wait for decades to create that reproductively viable coral. We can do it in a couple of years. We outplant these corals in a very similar way using that monoclonal outplant cluster design. Here we take the discs that came from the same parent colony with the same DNA, and we outplant them on the reef structure, basically attaching the ceramic plug onto the reef using epoxy. Now these corals will sheet out over the disc onto the reef structure, touch, fuse, and then start growing up. And here's a great image of what that looks like for the elkhorn coral. Now, again, instead of taking decades, it takes probably about 2 to 3 years after outplanting some of these slower growing species onto the reef to get them to be reproductively viable, which is our ultimate goal. Now, our land-based coral nursery, as I said, is the largest in the world. We hold about 30,000 microfragments or large-sized corals within any given time. We have thousands of different genotypes that we harvest, grow, and reproduce. And we outplant about 15,000 of these land-based corals every year. So we're hitting about 35,000 outplants every year. And I've been at Mote for about a decade. We are on an exponential increase trajectory. So we're constantly trying to think about bottlenecks, overcoming and upscaling our production every single year so that we can get all of these corals that need to be out on the reef back to their home. And I mentioned we have really good success after 1 year. We're starting to gather information now, 3 years, 5 years, even longer of some of our outplant sites. This is one of our most beautiful showcase reefs that I like to show an image of what success looks like. If you can imagine, this reef had no coral on it whatsoever, nothing. 3 years ago, after we started outplanting there, this coral reef has now 700% more coral on it. And you can just see how it's attracting the other wildlife in the area. There's fish surrounding these corals. There's other benthic invertebrates that are thriving just from putting corals out there. And we're getting over 99% survival on the cluster level. That means that we may lose a coral colony here or there, but the clusters are fusing and they're surviving and they're reproducing. And this has been a really nice way to showcase not just the short term, but the long-term impacts of these coral restoration efforts. I want to also emphasize that we are not just putting corals out there and crossing our fingers and hoping for the best. We know that environmental stress is going to continue to affect these corals, and many issues like ocean acidification, climate change, and diseases are things we're going to have to be battling for decades to come. But a lot of my research from my graduate school to when I came to Mote focused on understanding how different corals respond to stress. And some indeed are more resistant. And this is a picture of a couple corals in St. John in the U.S. Virgin Islands that I monitored every single month for 3 years. And they gave me the inspiration, which has led to a lot of our research here at Mote. So if you see, these are 2 elkhorn corals. There's a genotype on the left, we call it Genet 1, a genotype on the right, Genet 2. They look very similar right now. In fact, it's really difficult to tell that there's actually 2 corals here because the environmental conditions are very happy and healthy. But in the summer of 2005, it was one of the warmest water temperatures ever seen within our region for the past several decades. And so the water temperatures increased about 2 degrees Celsius above what they normally were. And this caused a different physiological response between these 2 genotypes because of the DNA that they carry. So the one on the left mainly retains its coloration, and the one on the right basically turns white. Now, this is a stress response that the coral exhibits when they undergo things like high water temperatures. Corals are animals, but they actually have little symbiotic algae that live inside of their tissue. That algae provides them with a bunch of nutrients that they need to survive. And then when they get stressed out, whether it's temperature or changes in salinity, they actually spit those algae out of their tissue and starve to death. Now, we want corals that don't do that. We want corals that can retain their algae and make sure that they survive those stressful conditions, because stress isn't going away, unfortunately. And this can have really important implications for what corals survive these stressful events that we're seeing more frequently throughout the year, because we want to outplant and produce the coral on the left. We don't want to put all of our energy into the coral on the right. That's just going to die the next summer. So we need to study why that coral on the left is so resistant to things like high water temperature and utilize that knowledge for better restoration practices. We have an amazing system down in the Keys called our Climate and Acidification Ocean Simulator. It's an image of it on the left. We have these aquaria that can mimic water temperature, water ocean chemistry. You can add things like nutrients, you can add pathogenic diseases, and it will help you either add a stress to it or mimic future ocean conditions like 100 years from now. And you can use this system to screen the genotypes in production to find the ones that are most resistant to the threats that we know aren't going away. Some of the endemic diseases within the region, high water temperatures, and the changing ocean chemistry. You can see an example of a study that we did with these corals on the right. All of these corals were in the same environmental conditions. The one on the right is the one that's the most robust. The one on the left is basically dead. So we can screen the corals that we have in propagation and production and know what makes them tick. What makes them special? Should we increase the production of them? Or should we do something else? We can actually use them as parents for the next generation. So I mentioned that we want to outplant the corals to be reproductively viable, but corals still have a hard time right now going through the entire reproduction cycle out on the reef because there's still very few in between kind of all of our restorations. There's very few of them out there naturally without our help. And there's all these issues associated with fertilization and settlement onto a reef that's covered in algae. So we have a whole staff, which is led by Dr. Hannah Cook, that is dedicated to doing assisted sexual reproduction in our lab down in the Keys. So we can actually use the information we gather in our resistance screening to inform the reproduction team who should be mom, who should be dad next year as they go through that spawning cycle. So we actually bring corals into our lab, and you can see an image of one of our big tubs that we can fill with reproductively sized corals that we've created, and we can allow them to spawn in the lab. Now they spawn by releasing eggs and sperm into the water column in these balls of gametes. There's actually eggs and sperm inside each one of those balls. They're hermaphrodites, many corals. And corals are very finicky. They actually only reproduce one night out of the year. So this particular species, the staghorn coral, releases their eggs and sperm about the fourth night after the full moon in August, about 3 hours after sunset. Tell me about it. So imagine having to go out onto the reef and be there waiting for those corals to spawn, which is how people have been doing it for decades. But we've kind of tried to hedge our bets. We do a little bit of that, but we also bring the corals into the lab in our in-water nurseries, in our land-based nurseries, and then they spawn in these controlled containers. So you're looking now under the water up into kind of the corals that are above you to the surface of the water. And above that is a team of people just waiting to collect all of those balls of gametes so that we can mix the eggs and sperm ourselves. And we can actually control who's mom, who's dad, or we can put them all together and mix them together and just let kind of nature take its course. And we do all of that because we know, yes, we want to increase those robust traits into the offspring, but we also want to like allow nature to take its course and create new things that we couldn't even think of, right? Because there's so many stressors out there. We want to make sure we still have a really genetically diverse population that we're using for these restoration activities. So this is what it does look like when you go collect corals from the wild. So in addition to kind of all of this effort that we utilize for the resilience screening of the corals we have in propagation, we also know that the corals that are out there and still like thriving, which are honestly few and far between, but they must be pretty robust and resistant as well. So each spawning season, we'll get a team to go out to these really unique survivors. We cover them with these mesh tents with a little bucket on the top. And when the corals spawn, all of the gametes go to the bucket at the top and you can kind of collect them. But let me tell you, this is risky business because everything has to align. The weather has to cooperate, can't have any storms, and of course they all reproduce during hurricane season. Your boat has to work, you have to have your field ops, and you're working all night long out in the field. So there's just a lot of variables that have to come into a play for all this to work well. And sometimes we get it, and sometimes we don't. But to hedge our bets, we also have created what we call our in-water spawning nurseries. So here we actually set up, instead of growing corals for propagation, where we kind of snip them and keep them small, we grow them big. We grow them to be reproductively viable out in those trees. And then right before they're about to spawn, we bring them into the lab. We put them into these containers and then they spawn for us at night. And we can do it in a controlled environment, which makes things happen a lot more smoothly. And we also now have integrated what are called land-based spawning systems. So that's what these aquaria are on the right. These aquaria systems mimic the temperature, the sunlight, the moonlight, all the things needed for corals to cue in on creating eggs and sperm and then releasing them into the environment. So we can utilize these systems to make the spawning more predictive. We can actually have them spawn in the morning. So we flip the day-night cycle, and then they think it's nighttime when it's really morning, which allows our staff and all of our interns to not turn into zombies. And we can also spawn them out of season. So we have a team that is flooded with activities in August, September, sometimes in July. But the rest of the year, they're rearing and propagating their recruits, but they have to wait until the next spawning season to get new material. This allows us to make the corals think that it's August when it's actually Christmas, right? So we can kind of manipulate their biology by triggering environmental cues on our cycle and allow an influx of gametes and new babies throughout the year instead of just relying on that one time of year. So there's a lot of novel technology that we're trying to integrate into our reproduction and restoration pipeline. And we also use our Mote pioneered methodologies for this too. So we can take our corals and we can microfragment them and grow them and fuse them in the lab and put them into our spawning system. So this allows us to learn a lot about these corals that we're growing and propagating and figuring out which ones we want to cycle in to be parents this year. So it gives us a ton of control. And we're not just having to outplant the corals and once they fuse, collect them out in the reef because of all the risky business associated with that. We put them in the spawning systems and we get them to spawn on our time. And we make tons of new coral babies. Every single year, we make thousands of new corals of multiple different species. Some species we're still trying to figure out how they reproduce and when. Some species we know a ton about, and we have so many new genotypes, we don't know what to do with them all. We honestly offload them to partners around the state to kind of spread out the wealth, basically, and give other people a chance to utilize these corals for research or restoration in other areas of the state that we're not directly working with. And these have led to some significant scientific breakthroughs. So this is what spawning looks like. So there's the release of the egg and sperm bundles into the water column. And let me tell you, if you blink for longer than a few seconds, you miss it. So it is, that's it. That's it. It's done. It goes. And that's it. But this spawning coral right here is a beautiful video of showcasing how our work is going to lead to and is leading to successful reef restoration. So this coral is one of the first corals we created using the microfragmentation and fusion process. It is a slow growing boulder species, and there are very few of them left out on the reef tract. Now this coral and a community of them around them, they all spawned in synchrony, so that they all spawned at the same time, which is what we want during the time that they are supposed to do it. And these corals survived an outbreak of stony coral tissue loss disease, which is the deadliest coral disease outbreak that has ever been recorded in the world. They survived Hurricane Irma, which was a category 4 when it hit this site. The eye of the hurricane hit this site. And they survived major bleaching events, 2 bleaching events. Those bleaching events are the high temperature conditions that cause the corals to spit out the algae and turn white. This is a key example of how the resilience research we're doing and the outplant methodology leads to coral restoration and reproduction. And this made international news because it provides such a beacon of hope that everything we've been working for and these assumptions that we've been making about how to do it the right way translates to real world success. And this has been a breakthrough for coral restoration research globally. But we're also not just doing restoration. So one of the things that I wanted to mention in the beginning and forgot is that there's all this demise happening and there's no natural recovery taking place, which is why we're intervening at such a high level, right? If you leave things up to itself, we would not have corals or a coral reef for much longer on the Florida Reef Tract. But as we're doing restoration and we're outplanting corals, we're also rescuing corals from the reef tract to bring them into a safe haven, a Noah's Ark for coral reef species diversity and genetic diversity. This is one of my favorite places to go. This is our International Coral Gene Bank, which is out at Mote's Aquaculture Research Park. So if you go to Sarasota and take Fruitville out east, where a lot of our fish aquaculture really bloomed, we now have infrastructure that's our Noah's Ark for corals. We are collecting corals that are being kept in a safe haven within these, we call these our warehouse banks. So each of these raceways contain now about 50 corals at least. And those corals are there as a safe haven. So no matter what happens out on the reef, today, tomorrow, 100 years from now, we will have a bank of species diversity and genetic diversity of our endemic indigenous corals that we can use to conduct the restoration essential to make sure we have a functioning ecosystem for generations to come. But in addition to that, these corals aren't just sitting there and waiting. They're actually getting integrated into our spawning room, which is adjacent to the warehouse. So we now have 4 of these spawning systems just within this one building alone. And we can produce gametes year round from these corals that are there as our saving grace from disaster and regional extinction. They are now the parents that create the babies that we funnel down into our restoration activities down in the Keys. So they're there as a safe haven, but they're also producing the genetic diversity that we need to do the restoration appropriately. And we're doing things like mixing resilience. So we have some corals that were harvested prior to a major disease outbreak affecting the coral reef in the Keys, the stony coral tissue loss disease. We got corals that were harvested prior to the disease outbreak moving through. We call them naive corals. But we also have survivors. So after the first wave of the disease went through, we went out and got survivors. So now we cross survivors with naive parents to have that beautiful genetic diversity, but the robust resistant disease traits that we need. So we're kind of trying to think about novel ways to apply all of this information we're gathering to do the best restoration science possible and to get the recovery we need on the reef so that those ecosystem services are there, providing shoreline protection, the base of the economy, all those novel compounds for diseases, all those functions are there. And finally, we are a major partner of an initiative led by NOAA, the National Oceanic and Atmospheric Administration, called Mission Iconic Reefs. This is the largest multi-member restoration project that has ever been coordinated in the world. The goal is to restore these 7 reefs, which are all iconic reefs from the Keys. Ones that if you go to the Keys, that's where you're going to dive because those are the memorable experiences. Those are what used to be the thriving iconic reefs of the 80s and 70s and prior. The goal is to bring these reefs back to their previous coral cover, which is about 30% over the next 20 years. And we're one of only 2 major partners that are providing all the resources and the corals to make it all happen. And finally, we are expanding our operations throughout the Keys. So we're not just working on those 7 iconic reefs, although a lot of our effort is going into some of those. We're actually outplanting now at over 60 different reef sites throughout the entire Florida Keys every single day. So we have teams that go out to all of these different areas, kind of as much as possible to add coral, to add to the community, to bring back those coral reefs functionality to life. And we have kind of started spreading. You can see there's kind of a gap in the middle Keys. And within next year, that entire area is going to be covered by those dots, which are where we're actually outplanting. So we are expanding at an exponential rate as we're upscaling how we produce and integrate that restoration science. And just to kind of wrap up the big picture, these are all the steps that I talked about in the presentation today, from start to finish. And it is cyclical, and it's sustainable, and it's novel, and it all happens at Mote. This is the only organization in the world that can do the entire cycle that's essential for resilient reef restoration under one roof, that increases efficiency, that makes our productivity higher. We have a lot less red tape to jump through, and it is just an efficient powerhouse of production. And it's amazing that we have the capabilities to do it. And it's why we have so many people that are so passionate about coral reefs under one roof, is because it's an amazing cycle. It's the only way we're going to get Florida's coral reef back to some semblance of what it was decades ago. But it's also the passion that really brings us all together in order to have these corals for generations to come, because corals actually have an indefinite lifespan. So what we do today is not only going to influence the reefs of tomorrow or a year from now or a decade from now, it can live actually thousands of years. So everything we do today can have impacts for generations upon generations. And it's a really incredible privilege to be a part of that. And I just want to thank you so much for your time. I really appreciate the attention. And I just want to recognize that all of this work is a combination of philanthropy, federal support, state support, county support. It takes a huge village to do this. And we're incredibly grateful for community members like you coming to learn more about it, and funders like these groups helping us make the work possible. Thank you.